Hello everyone. Today we will see how isolation of bacteria present in the soil can be done. The most simplest and widely used method of isolation of soil bacteria is the serial dilution technique. The principle of the serial dilution technique is based on the stepwise dilution of a known quantity of soil sample in a known quantity of sterilized water. Due to serial dilution, the number of microorganisms gradually decline. The glasswares required for isolation of bacteria are conical flask, petri dishes, 1 ml pipette, and test tubes. The culture media used for isolation of bacteria is nutrient agar medium. The composition of nutrient agar medium is peptone 5 gram, sodium chloride 5 gram, beef extract 3 gram, agar 15 gram, and distilled water 1000 ml. The petri dishes, conical flasks, and other glasswares were washed and put in a hot air oven for sterilization at 160 degrees centigrade for 1.5 hours. After preparation of the nutrient agar medium, the conical flask is plugged with cotton and it should be sterilized in autoclave at 121 degree centigrade. Soil sample is collected aseptically by sterilized scalpel. The laminar airflow chamber is cleaned with cotton and alcohol. Sterilized glasswares, culture medium, distilled water is put inside the chamber and UV light is switched on for 5 minutes. For preparation of the dilution, 4 test tubes are taken and 10 ml distilled water is poured in the first test tube and rest test tube are poured with 9 ml of distilled water and they are marked as 1 to 4. Now 1 gram of soil is put in the first test tube and mixed thoroughly by shaking. Then 1 ml of this solution is transferred into the second test tube and dilution is marked as 10 to the power minus 1. From the test tube number 2, 1 ml of solution is transferred to the test tube number 3 and dilution is marked as 10 to the power minus 2. Similarly, from the test tube number 3, 1 ml of solution is transferred to the test tube number 4 and dilution is marked as 10 to the power minus 3. Here 10 to the power minus 3 dilution is taken for isolation of bacteria. Now 3 pairs of petri dishes is taken for isolation of bacteria. 1 ml of inoculum from the 10 to the power minus 3 dilution is poured in the petri dishes. In the 3 pairs of petri dishes, the sterilized nutrient agar medium is poured in such a way that the medium is uniformly spread in the petri dishes. Then petri dishes are rotated smoothly for uniform distribution of inoculum, and all these works are done under the laminar airflow chamber over flame. After solidification of the media, all the petri dishes are kept in a incubator at 30 degrees centigrade for 48 hours. After the specific incubation period, the petri dishes are taken out and the colonies were counted. If the total number of CFU is multiplied with reciprocal of the dilution factor, then we can get the total bacterial count per gram of soil. Here we have taken 10 to the power minus 3 dilution, and if the total number of CFU is 100, then 100 should be multiplied with reciprocal of 10 to the power minus 3. Then the total bacterial count per gram of soil will be 100 into 10 to the power 3.